Welcome to the Deep Sleep Discoveries channel. Tonight, we drift into the vast and mysterious world of marine biology. These ocean facts are not meant to be memorized, only felt. 100 gentle truths about sea life, deep oceans, and hidden ecosystems designed to slow your thoughts and carry you softly towards sleep, guided by the quiet science of the sea. Before we begin, please consider liking and subscribing to the channel. It helps others find their way here too. Now, settle in and relax. We begin near the surface with familiar rhythms of water and light, then slowly descend together into deeper waters where attention softens, curiosity quiets, and the first gentle fact waits below in calm, unhurried time now. Marine biology is the science devoted to understanding life in salt water, from shallow shores to the deepest basins. It examines animals, plants, microbes, and entire ecosystems, asking how they survive, interact, and change over time. Because the ocean covers most of Earth, marine biology quietly studies most of the planet's living space. It connects chemistry, physics, geology, and biology into one slow conversation shaped by tides and currents. This field is not limited to famous animals like whales or sharks. It also follows drifting plankton, sediment-dwelling worms, and microbes that reshape water chemistry. Marine biologists observe, sample, model, and listen, often working in environments where visibility is low and patience matters. Progress can be gradual, built from long records rather than quick discoveries. Together, these efforts reveal patterns that link distant coasts and deep waters into one system. As this story widens, it naturally leads from the science itself to the setting it studies most, the vast, interconnected ocean that covers our world and quietly shapes climate, weather, breath, food, and time itself together endlessly. Oceans cover more than 70% of Earth's surface, yet their importance goes far beyond area alone. They contain nearly all habitable volume on the planet, offering room for life not just across distance, but through depth. From sunlit surfaces to cold trenches, the ocean stacks habitats vertically, creating layers where different organisms thrive. This vastness allows marine life to spread risk and opportunity. A change in one region can ripple outward through currents, migrations, and chemistry. Heat absorbed by surface waters moves slowly around the globe, shaping climate patterns far from the sea. Nutrients lifted from the deep can fertilize surface food webs, feeding creatures large and small. Because of this scale, the ocean acts like Earth's main life support system. It moderates temperature, stores carbon, and sustains cycles that unfold quietly over centuries. Understanding that immensity prepares us to look closer, shifting from sheer size to how the ocean organizes itself into zones defined by light and depth, which set the stage for life below the surface next, where sunlight fades, pressure rises, and adaptation begins in earnest, quietly, patiently, without pause anywhere else. The ocean is organized into zones defined by light, depth, and pressure, rather than borders you can see. Near the surface, sunlight drives photosynthesis and fuels busy food webs. As depth increases, light thins, temperatures fall, and pressure climbs steadily, reshaping what bodies can safely contain. These zones are not rigid walls, but gradual shifts. Animals move between them daily or seasonally, following food and safety. Some rise toward the surface at night, then sink again before dawn. Others remain adapted to narrow bands, finely tuned to specific conditions of darkness and force. Pressure itself does not crush most deep-sea life because their bodies are largely water, with internal pressure matching the outside. What disappears instead are gas-filled spaces which cannot survive the squeeze. Each zone filters possibilities, favoring certain shapes, senses, and strategies. Understanding this layered structure helps explain why life changes with depth. To see how energy enters this system at all, we begin at the very top, where sunlight still reaches the sea and quietly sets limits on abundance, motion, rhythm, everywhere. Sunlight reaches only the uppermost layer of the ocean, known as the photic zone. This thin skin of water, compared to the ocean's full depth, is where light is strong enough to power photosynthesis. Here, microscopic phytoplankton capture energy from the sun and turn it into living matter, forming the base of most marine food webs. Because light fades quickly with depth, Life in the photic zone is shaped by brightness, shadow, and daily cycles. Animals often time their movements to sunrise and sunset, balancing feeding opportunities with the risk of being seen. The surface can appear calm, but beneath it, constant motion unfolds as currents mix nutrients upward and organisms respond. This narrow layer matters far beyond its size. It feeds the deep, fuels oxygen production, and connects the ocean to the atmosphere above. Once sunlight disappears, life must find other ways to survive, which leads naturally downward into the darker layers where energy arrives indirectly and adaptation becomes quieter and more specialized. Many fish do not hear with ears like ours, but with bodies tuned to vibration. Pressure waves moving through water pass easily into tissue, allowing fish to sense sound as movement rather than noise. Some fish detect vibrations through their swim bladders, which can amplify sound, while others rely on sensory systems along their sides. These signals help them locate predators, find mates, and orient within reefs or schools. In low-visibility environments, vibration can provide early warning. A sudden pressure change may signal an approaching predator or a shifting current. Fish respond instantly, often without conscious processing, guided by evolved sensitivity. Marine biologists record underwater soundscapes to learn which species are present and active. Reefs in particular can sound busy with clicks, hums, and pulses. This hidden acoustic world reveals that even seemingly quiet waters are alive with information. From here, attention turns to habitats where structure and sound combine, beginning with coral reefs built by animals themselves. Coral reefs may look like colorful rock formations, but they are built by living animals. Each coral structure begins with tiny coral polyps, soft-bodied creatures related to jellyfish. These polyps secrete hard skeletons made of calcium carbonate beneath themselves, and over time, generations stack these structures into ridges, shelves, and towers. Although individual polyps are small and fragile, together they create massive habitats that support thousands of species. Fish shelter in the crevices, invertebrates cling to surfaces, and predators patrol the edges. Reefs become neighborhoods shaped by patience rather than speed. Because corals grow slowly, reefs record time in layers. A healthy reef may take centuries to form and only moments to damage. Marine biologists study coral growth to understand both resilience and vulnerability. To survive in clear, nutrient-poor waters, corals rely on an important partnership, one that quietly powers reef life and leads directly to the microscopic allies living inside coral tissues. Most reef-building corals live in close partnership with microscopic algae that reside inside their tissues. These algae use sunlight to produce energy through photosynthesis and share much of that energy with the coral host. In return, the coral provides shelter and access to light. This relationship allows corals to thrive in clear tropical waters where nutrients are scarce. It also gives reefs their vivid colors. When conditions remain stable, this partnership fuels growth and reef expansion. However, the balance is delicate. Prolonged stress, especially from warmer water, can cause corals to expel their algal partners. Without them, corals lose color and energy, entering a weakened state known as bleaching. Recovery is possible if conditions improve, but prolonged stress can be fatal. Marine biologists monitor these partnerships closely because they reveal how environmental change affects entire ecosystems. Understanding coral-algae cooperation sets the stage for exploring how reefs reproduce, renew themselves, and synchronize life with the rhythms of the ocean and sky. Many corals reproduce by releasing eggs and sperm directly into the water in events known as spawning. These releases often occur simultaneously across large sections of reef, filling the water with drifting bundles of reproductive cells. Timing is critical. Corals often synchronize spawning with seasonal temperature changes, tides, and moonlight. By releasing together, they increase the chances that eggs will meet sperm while overwhelming predators with sheer abundance. After fertilization, coral larvae drift in the plankton before settling onto suitable surfaces. Only a small fraction survive long enough to attach and begin building new reef structures. This makes successful spawning events essential for reef renewal. Marine biologists plan carefully to observe these brief windows, which may occur only once a year and last a single night. Spawning highlights how reef life depends on coordination rather than individual success. Once larvae settle, they must compete for space, light, and safety, leading into the crowded, dynamic communities that define reef ecosystems next. Reef fish display a remarkable range of colors and patterns that serve purposes beyond beauty. Color can signal aggression, submission, readiness to mate, or a shift in social role. Some changes occur gradually, while others happen in moments. Pigment cells in fish skin expand or contract, altering appearance without changing the underlying structure. In some species, color patterns intensify during territorial disputes or fade at night to reduce visibility. Color also aids camouflage. Many fish match the hues and textures of coral, sand, or shadow, blending into complex backgrounds. This reduces detection by predators and allows fish to approach prey unnoticed. Marine biologists study these changes to understand how communication works without sound or scent. On a reef, color becomes a language shaped by light and motion. These visual signals interact with other behaviors, including cooperation, cleaning, and mutual aid, which appear clearly in the relationships between different reef species. On coral reefs, cleaner fish establish stations where larger fish gather to have parasites and dead tissue removed. These interactions temporarily suspend the usual predator-prey rules. Clients hold still, sometimes even opening their mouths while cleaners work. For cleaner fish, the benefit is a steady food source. For the clients, reduced parasite loads improve health, mobility, and survival. This mutualism benefits both sides and can influence which fish thrive in crowded reef environments. Reputation matters. Cleaners that cheat by biting healthy tissue may lose clients, while reliable cleaners attract repeat visits. Some fish even queue patiently, waiting their turn. Marine biologists study cleaning behavior to understand cooperation in competitive ecosystems. These stations become social hubs where trust and benefit intersect. From these partnerships, attention naturally moves to larger reef inhabitants whose presence shapes entire ecosystems through appetite and influence. Sharks play important roles in reef ecosystems, not only through what they eat, but through how their presence alters behavior. Fish may avoid certain areas or adjust feeding patterns when sharks are nearby, reducing overgrazing in sensitive zones. This influence extends beyond direct predation. Fear alone can reshape how energy moves through a reef, distributing pressure more evenly. In places where sharks decline, prey species may grow bolder, sometimes leading to imbalances that affect coral health. Marine biologists study these dynamics to understand top-down control in marine food webs. Sharks help maintain diversity by preventing any one species from dominating. Their role reminds us that ecosystems depend on relationships, not just numbers. From large predators, focus shifts to quieter architects of reef environments, especially organisms that build and reshape habitats beneath the surface. Kelp forests grow in cooler, nutrient-rich waters and form towering underwater canopies. Some kelp species can grow over a foot per day, transforming bare seafloor into dense habitat quickly. These forests provide food, shelter, and nursery space for many marine animals. Fish weave through fronds, invertebrates cling to blades, and predators hunt along the edges. Kelp also slows water movement, reducing wave energy and coastal erosion. Because kelp depends on sunlight and nutrients, it responds quickly to environmental change. Warm water or loss of grazers' predators can cause forests to decline rapidly. Marine biologists view kelp as both resilient and fragile. Their rapid growth offers recovery potential, but imbalance can undo them just as fast. This leads naturally to animals that strongly influence kelp survival, particularly those that graze relentlessly when left unchecked. Sea otters are powerful influences in kelp forest ecosystems. They feed heavily on sea urchins, which otherwise graze kelp down to bare rock. When otters are present, urchin populations remain controlled, allowing kelp to grow tall and dense. This relationship creates cascading effects. Kelp forests provide habitat for fish and invertebrates, which support birds and larger predators. Without otters, kelp can disappear, reducing biodiversity and altering coastal ecosystems. Marine biologists describe otters as keystone predators because their impact is disproportionately large compared to their numbers. One species can shape an entire habitat. Otters also display unique behaviors, such as using rocks as tools to crack shells. Their influence connects feeding, behavior, and habitat structure. From kelp forests, attention shifts shoreward, where land and sea blend in habitats shaped by roots and tides. Mangroves grow where land meets sea in warm regions, forming dense root networks that slow water and trap sediment. These tangled roots create sheltered spaces where young fish and crustaceans can hide from predators. Mangroves also protect coastlines by reducing wave energy and stabilizing shorelines during storms. Leaf litter from mangroves feeds microbial communities that support food webs extending offshore. Because mangroves sit at the boundary between environments, they link land-based nutrients to marine ecosystems. Changes upstream can directly affect mangrove health and the species that depend on them. Marine biologists value mangroves as nurseries that support fisheries and biodiversity. Their loss can ripple outward, weakening nearby reefs and seagrass beds. From these rooted forests, the focus moves underwater again to another quiet but vital coastal habitat. Seagrass meadows are underwater flowering plants that form broad fields in shallow, calm waters. Unlike seaweed, seagrass has roots, stems, and leaves adapted to life beneath the surface. These meadows slow currents, trap sediment, and create stable environments for small animals. Fish, crabs, and grazers rely on seagrass for food and shelter, especially during early life stages. Seagrass also plays a powerful role in storing carbon. Organic material becomes buried in oxygen-poor sediments beneath the plants, where it can remain locked away for centuries. This makes seagrass an important ally in regulating climate. Marine biologists study seagrass for both ecological and climate reasons. Though it may appear simple, a meadow functions as a living filter, nursery, and carbon vault. From here, attention widens toward broader coastal systems where freshwater and saltwater mix and productivity peaks. Coral bleaching occurs when corals lose their microscopic algal partners after prolonged stress, most often from elevated water temperatures. The algae normally provide much of the coral's energy, so their loss leaves the coral pale and weakened, but not immediately dead. Bleaching is a survival response, not an instant collapse. If conditions improve soon enough, algae can return and the coral may recover. If stress continues, the coral can starve, making reefs vulnerable to erosion and takeover by algae. Bleaching can unfold across vast areas, transforming colorful reefs into quiet white landscapes within weeks. Marine biologists track bleaching because it reveals how tightly reef health is tied to temperature stability. Even small, sustained changes can disrupt relationships built over thousands of years. Bleaching does not remove the reef structure right away, but it reduces the living layer that maintains it. Understanding bleaching prepares us to see reefs as dynamic systems rather than static scenery. From stress and recovery, attention turns naturally to how reefs persist over time despite slow growth and repeated disturbance. Coral reefs grow at a pace that is almost imperceptible on human timescales. Most reef-building corals add only millimeters to centimeters per year as they deposit calcium carbonate skeletons beneath themselves. Over centuries, these small additions accumulate into massive structures. Because growth is slow, reefs store history. Layers of skeleton record past conditions, including temperature, water chemistry, and storm events. Damage that happens quickly can therefore erase centuries of construction in a single season. Marine biologists study growth rates to understand resilience. Some reefs recover after disturbance if conditions remain favorable, while others struggle if repeated stress interrupts rebuilding. Growth also depends on balance. Grazers must keep algae in check, and water quality must remain clear enough for light to reach coral tissues. Reefs are neither fragile ornaments nor indestructible fortresses. They are patient systems built by countless tiny efforts repeated over time. Recognizing that patience leads into the remarkable timing strategies reefs use to ensure the next generation can continue building when conditions allow. Many reef organisms synchronize reproduction with the lunar cycle. On certain nights, often linked to full moons and specific tides, corals and other invertebrates release eggs and sperm simultaneously into the water. These events can turn the sea into a drifting cloud of potential life. Moonlight and tidal timing help coordinate spawning across large areas, increasing fertilization success. Releasing together also overwhelms predators, allowing more reproductive cells to survive simply through abundance. The event may last only minutes, yet its impact can shape reefs for decades. Marine biologists plan years in advance to observe these nights because missing them means waiting another full cycle. Spawning demonstrates how life uses predictable celestial rhythms as a calendar. After fertilization, larvae drift with currents before settling, facing long odds and open water. This moment links the sky to the sea and chance to persistence. From these synchronized beginnings, reef communities grow crowded and competitive, leading to the colorful interactions among reef fish that follow. Reef fish often live within structured social groups shaped by territory, size, and dominance. Some species defend small patches of reef, while others move freely through shared space. These arrangements reduce conflict and help distribute resources in crowded environments. Hierarchy plays a key role. Larger or more dominant individuals may control access to food or breeding opportunities, while smaller fish wait or adopt alternative strategies. Social order can shift quickly if a dominant fish disappears. Marine biologists study these systems to understand how behavior stabilizes ecosystems. Social structure influences feeding pressure, reproduction, and survival. It also explains why removing certain individuals can have outsized effects on reef balance. Communication within these groups relies on color, posture, and movement rather than sound. A subtle change in hue or fin position can signal submission or challenge. These silent conversations maintain order in busy habitats. Understanding reef social life prepares us to explore one of its most striking features: the ability of some fish to change their biological role entirely when the group requires it. Some reef fish can change sex in response to social conditions, a process known as sequential hermaphroditism. This is common in groups like wrasses and clownfish, where reproductive roles depend on hierarchy rather than fixed anatomy. In a clownfish colony, one dominant female leads, followed by a breeding male and smaller non-breeders. If the female dies, the largest male undergoes a physical transition into a female and another fish becomes the breeding male. This shift involves hormonal changes and restructuring of reproductive organs over time. Marine biologists study this because it shows how flexible biology can be when shaped by social need. Sex change stabilizes reproduction in environments where finding new mates is uncertain. This strategy challenges assumptions about fixed roles and highlights how evolution favors systems that preserve continuity. From flexible bodies, attention turns to flexible behaviors, especially those involving cooperation between different species that share reef space in surprising ways. Mutualism is common on reefs, where different species benefit from close interaction. Cleaner fish and shrimp remove parasites from larger fish, gaining food while improving their clients' health. Both partners gain survival advantages without direct competition. These relationships rely on trust and repetition. Clients return to reliable cleaners, and cleaners that cheat risk losing future visitors. Some species even signal willingness to be cleaned through posture and color changes. Marine biologists see mutualism as a stabilizing force in dense ecosystems. It reduces disease, supports growth, and allows species to specialize rather than compete for the same resources. Mutualistic interactions also extend beyond animals. Corals rely on algae, and some fish shelter among stinging anemones in exchange for protection and nutrients. These exchanges form invisible threads that hold reef systems together. Recognizing cooperation as a survival strategy leads naturally to predators whose influence is felt not through partnership, but through presence, shaping reefs by altering how other animals behave. Large predators affect reef ecosystems even when they are not actively hunting. Their presence alone can change how prey animals move, feed, and rest. Fish may avoid open areas or graze more cautiously when predators are nearby. This behavioral influence spreads pressure across the reef, preventing intense grazing in one location. As a result, corals and algae experience more balanced conditions. When predators decline, prey species may become bolder, sometimes leading to ecosystem imbalance. Marine biologists describe this as a landscape of fear where risk shapes decisions. The effect is subtle but powerful, guiding daily routines without constant predation. Predators act as regulators of behavior as much as population size. Their influence demonstrates that ecosystems are built from choices, not just consumption. From reefs shaped by fear and movement, attention shifts to other coastal habitats where structure, not predators, provides safety for young marine life. Kelp forests create three-dimensional structure in coastal waters, rising from the seafloor towards sunlight. Their long blades sway with waves, forming corridors, shadows, and shelter that transform open water into habitat. This architecture supports many species at once. Small fish hide among fronds, invertebrates cling to surfaces, and predators patrol edges. The vertical structure allows animals to occupy different layers without direct competition. Kelp also modifies physical conditions. It slows water movement, dampens wave energy, and reduces coastal erosion. These effects create calmer zones where young animals can survive their most vulnerable stages. Marine biologists study kelp forests as examples of living structure. Like coral reefs, they show how organisms can shape environments rather than simply occupy them. Their fast growth contrasts sharply with coral patience. This contrast highlights the importance of animals that regulate kelp grazers, leading directly to the role of sea urchins and their predators next. Sea urchins feed by scraping algae and kelp from hard surfaces using specialized mouthparts. In balanced systems, their grazing helps prevent algae from overwhelming reefs and rocky bottoms. When unchecked, however, urchins can consume kelp faster than it can regrow. This overgrazing can create urchin barrens, areas of bare rock with little habitat complexity. These zones support fewer species and reduce coastal productivity. Marine biologists study urchin populations to understand tipping points. Small changes in predator presence can allow urchin numbers to explode, reshaping entire ecosystems. Urchins themselves are not destructive by nature, their impact depends on balance. When predators return, grazing pressure eases and kelp can recover. This back and forth highlights how tightly linked marine systems are. From grazers that shape forests, attention moves shoreward again to habitats where roots, sediment, and water movement combine to protect young life before it reaches the open sea. Estuaries form where freshwater rivers meet the sea, creating environments where salinity shifts with tides and seasons. These changing conditions may seem harsh, but they support some of the most productive habitats on Earth. Nutrients from rivers mix with marine waters, fueling plankton growth. Shallow depths warm quickly, accelerating development for young fish, crabs, and shrimp. Mudflats, marsh plants, and submerged grasses provide shelter from predators. Many marine species depend on estuaries during early life stages before moving offshore as adults. This makes estuaries essential to fisheries and food webs far beyond their boundaries. Marine biologists call estuaries nurseries because survival rates are higher there than in open water, yet they are sensitive to pollution, runoff, and physical alteration. Changes upstream are felt quickly downstream. Understanding estuaries reveals how tightly land and ocean are connected. From these mixing zones, the story naturally widens toward the open sea again, where vast processes operate beyond shorelines. Salt marshes grow along sheltered coastlines where tides regularly flood and drain grassy flats. The plants are adapted to salt, shifting water levels, and oxygen-poor mud, creating habitats that appear calm but work constantly. As water moves through marsh grasses, suspended particles settle out, and microbes in the sediment process excess nutrients. This filtering improves water quality for nearby bays and coastal seas. Marshes also act as natural buffers during storms. Dense vegetation slows incoming waves and storm surges, reducing erosion and flooding inland. The marsh absorbs energy that would otherwise strike harder surfaces. Many fish and crustaceans use marshes as nurseries, finding food and shelter during early life stages. Birds rely on these areas for feeding and nesting. Marine biologists study marshes because they sit at the boundary between land and sea, responding quickly to changes upstream. When marshes are lost, coastlines become more vulnerable and water quality declines. From these grassy edges, attention moves to broader coastal systems shaped by temperature, salinity, and water movement together. Coastal ecosystems do not exist in isolation. Mangroves, salt marshes, seagrass beds, reefs, and open water exchange nutrients, organisms, and energy continuously. A change in one habitat often echoes through others, even when they appear separate. For example, mangroves trap sediment that might otherwise smother reefs. Seagrass stabilizes the seabed, improving water clarity for nearby corals. Estuaries supply young fish that later populate reefs and offshore waters. These links form networks rather than linear chains. Marine biologists study these connections to understand ecosystem resilience. Protecting one habitat can strengthen others, while damage in one place can weaken the whole system. This perspective shifts conservation from isolated sites to regional planning. Coastal connections also blur the line between land and sea. Rainfall, river flow, and human activity upstream influence marine life downstream. Understanding connectivity prepares us to move outward again, away from shore, into open ocean systems where movement and migration dominate life patterns. The open ocean, far from coasts and reefs, can appear empty at first glance. Yet it hosts a wide range of life adapted to vast distances and sparse resources. Food is often scattered, so animals must travel efficiently and sense opportunity over long ranges. Many open ocean species are strong swimmers or drifters shaped by currents. Tuna, sharks, and whales roam across entire basins following seasonal productivity. Smaller organisms, including plankton and jelly-like animals, form layers that rise and sink daily. Because there are few physical landmarks, the open ocean relies on gradients. Temperature, salinity, and chemistry guide movement more than place. Marine biologists study these systems using satellites, tags, and models rather than fixed observation points. Life here depends on timing as much as location. Productivity blooms briefly, then fades, requiring flexibility. From this wide, mobile environment, attention turns to one of the ocean's most predictable movements: daily vertical migration beneath the surface. Every day, enormous numbers of marine animals move up and down through the water column in a pattern called daily vertical migration. As night falls, small fish, crustaceans, and jelly-like animals rise toward the surface to feed. Before sunrise, they sink back into deeper, darker layers. This movement is driven by a balance between hunger and safety. Darkness provides cover from predators, allowing animals to access surface food with less risk. Daylight sends them retreating to deeper water where shadows offer protection. In sheer number, this migration is considered the largest animal movement on Earth. It shifts nutrients, redistributes energy, and plays a role in moving carbon deeper into the ocean. Marine biologists track this migration with sonar, which reveals rising and sinking layers of life. These daily pulses shape food availability for larger predators. From movement and timing, focus shifts toward animals whose migrations span not just depth, but entire oceans over months and years. Many marine animals migrate across thousands of miles during their lifetimes. Whales travel between feeding grounds and breeding areas. Sea turtles cross entire oceans to return to natal beaches. Fish follow temperature bands and productivity zones seasonally. These migrations rely on multiple cues. Some animals use Earth's magnetic field as a map. Others follow chemical signals, currents, or memory shaped by experience. Migration allows species to exploit resources that shift with seasons. Marine biologists study migration through tagging, tracking, and genetics. Understanding routes helps identify critical habitats that need protection, such as breeding grounds and feeding hotspots. Migration connects distant ecosystems. What happens in one region can affect populations half a world away. Disruptions along a route can echo across generations. From large-scale journeys, attention narrows again to animals that remain closer to home, using remarkable physiological adaptations to survive in specific environments rather than traveling away from them. Marine mammals evolved from land ancestors and had to solve the challenge of breathing while living underwater. Whales, seals, and dolphins manage oxygen carefully to support long dives. They store oxygen not only in their lungs, but also in blood and muscles. During dives, heart rate slows, and blood flow is prioritized for essential organs like the brain and heart. Non-essential tissues temporarily receive less oxygen. This strategy allows dives lasting minutes or even over an hour in species like sperm whales. Marine biologists study these adaptations using tags that record depth, heart rate, and movement. Efficient oxygen use also reduces nitrogen absorption, lowering the risk of decompression problems. These systems reflect precise control rather than brute endurance. Understanding marine mammal physiology helps explain their vulnerability to disturbance. Stress or forced rapid surfacing can disrupt carefully balanced processes. From breadth and depth, attention turns to sleep, a necessity even in an environment that never truly rests. Some marine mammals can sleep with only half of their brain at a time, a state known as unihemispheric slow-wave sleep. This allows one hemisphere to rest while the other remains alert. In fur seals, this ability is especially common at sea. One side of the brain monitors breathing, posture, and potential danger while the other sleeps. The resting side alternates, allowing full recovery over time. This form of sleep helps animals remain afloat and responsive in environments where complete unconsciousness could be risky. Marine biologists study unihemispheric sleep to understand how rest adapts to survival needs. Unlike human sleep, this process is flexible and situational. Seals may sleep differently on land than at sea, adjusting to risk levels. From sleep adaptations, focus shifts to animals that rely less on vigilance and more on armor, regeneration, and unusual defenses to survive encounters in the marine world. Many marine animals possess the ability to regenerate lost or damaged body parts. This includes regrowing limbs, tissues, and even complex structures over time. Sea stars can regrow arms, sometimes regenerating entire bodies from a single limb. Sea cucumbers can expel internal organs to escape predators, then slowly rebuild them. These processes require precise cellular coordination. Regeneration is not fast. It demands energy and leaves animals vulnerable during recovery. Yet in environments where injury is common, the ability to rebuild can mean survival rather than death. Marine biologists study regeneration to understand how cells know where to grow and when to stop. Insights from these animals inform broader biological questions about healing and development. Regeneration reflects patience rather than resilience through force. From rebuilding bodies, attention turns to creatures that avoid damage altogether through intelligence, camouflage, and complex behavior. Cephalopods such as octopuses, cuttlefish, and squid display remarkable intelligence among invertebrates. They solve problems, use tools, and learn from experience. Octopuses can open containers, navigate mazes, and manipulate objects with flexible arms equipped with chemical sensors. Their nervous systems are distributed, allowing arms to act semi-independently while remaining coordinated. Marine biologists study cephalopod intelligence to understand alternative paths to cognition. Unlike vertebrates, these animals evolved intelligence without a backbone, showing that complex behavior can arise in different forms. Camouflage, memory, and curiosity help cephalopods survive short lifespans in dangerous environments. Intelligence compensates for vulnerability. Their behavior highlights a theme common in the ocean: survival through adaptation rather than dominance. From intelligent hunters, focus shifts to communication methods that rely not on sound but on light and color in ever-changing displays. Many marine animals communicate through rapid color change. Squid and cuttlefish use specialized skin cells to produce waves of color, contrast, and pattern across their bodies. These displays can signal aggression, courtship, coordination, or warning. Changes happen in fractions of a second, allowing responses to shifting social situations. Color becomes a language written on the body itself. Marine biologists study these signals by recording behavior and matching patterns to context. Communication often occurs without sound, especially in environments where noise or distance makes acoustics unreliable. Color change also supports camouflage. Animals can blend into backgrounds or disrupt outlines to confuse predators. The same system supports both concealment and expression. This dual use reflects efficiency. One biological tool serves multiple purposes depending on need. From visual communication, the story naturally continues toward the deep ocean again, where light fades and life must create its own illumination to be seen at all. In the deep ocean, where sunlight never reaches, many animals produce their own light through bioluminescence. This light is created by chemical reactions inside the body or by glowing bacteria living in specialized tissues. The result is a brief flash, steady glow, or patterned signal in otherwise total darkness. Bioluminescence serves many purposes. Some animals use light to lure prey closer. Others flash warnings or confuse predators with sudden brightness. Certain species match the faint glow from above to erase their silhouette, a form of camouflage known as counter-illumination. Marine biologists study bioluminescence using low-light cameras and submersibles because bright lights can alter natural behavior. The deep sea is not empty or silent. It is filled with signals that appear and vanish in moments. Light becomes language, defense, and deception at once. As depth increases, vision narrows, and light becomes precious. From glowing bodies, attention shifts to animals that manipulate darkness further, sometimes using light not to reveal themselves, but to misdirect danger entirely. Some deep-sea animals use bioluminescence as a distraction rather than a beacon. When threatened, they release glowing secretions or shed luminous body parts that continue shining as they escape into darkness. The predator's attention follows the light, not the animal. This strategy is especially useful in open water, where there are no rocks or crevices to hide within. Light becomes a temporary cover, buying seconds that can mean survival. In some species, detachable glowing tips wriggle after release, further holding focus. Marine biologists study these behaviors to understand predation in environments where concealment is rare. In the deep sea, attention is currency. Whoever controls it often survives. These decoys demonstrate that light in the ocean is not always about communication. Sometimes it is misdirection. From animals that create and abandon light, focus shifts toward creatures that do not glow at all, instead relying on chemistry and pressure to survive extreme depth quietly. At great depths, pressure increases dramatically, yet many animals thrive there. They are not held together by pressure, but balanced by it. Because their bodies are largely composed of water, which does not compress, internal pressure equals the pressure outside. What deep-sea animals lack are gas-filled spaces. Structures like swim bladders would collapse, so these animals rely on neutral buoyancy and soft tissues instead. Enzymes and membranes are adapted to function under constant force. Marine biologists study pressure adaptation by bringing samples up carefully or recreating deep conditions in specialized chambers. Removing pressure too quickly can damage tissues that evolved to expect it. Life here moves slowly, energy is conserved, movement is deliberate. Pressure becomes a constant presence rather than a threat. Understanding pressure adaptation reveals how life solves physical constraints without resistance. From pressure-tolerant bodies, attention turns to chemical extremes where life survives not by structure alone, but by feeding on compounds toxic elsewhere. In parts of the deep ocean, life exists without any connection to sunlight. Around hydrothermal vents and cold seeps, microbes form the base of ecosystems using chemosynthesis instead of photosynthesis. They convert chemical energy into food. These microbes use compounds like hydrogen sulfide or methane released from the seafloor. Larger animals rely on them directly or indirectly for nourishment. Entire food webs grow around these chemical sources. Marine biologists were surprised by these discoveries because they revealed that sunlight is not required for complex ecosystems. Life can be powered by geology alone. Vent communities can appear and disappear as geological activity shifts. Species must disperse and colonize new sites, making survival dependent on timing and chance. Chemosynthesis expands the definition of habitability on Earth. From chemical energy, focus moves to animals that form some of the most extreme partnerships in the ocean, hosting microbes inside their own bodies to survive in places where food floats nowhere else. Giant tube worms near hydrothermal vents lack mouths and digestive systems. Instead, they host bacteria inside a special organ. These bacteria perform chemosynthesis, turning vent chemicals into food that nourishes the worm. The worm provides oxygen and sulfide through its blood, carefully regulating both to keep its bacterial partners productive. In return, the bacteria supply all necessary nutrients. The worm becomes a living vessel for another life form. Marine biologists study this partnership because it shows how animals can outsource digestion entirely. What appears to be a single organism is actually a tightly managed community. Tube worms can grow large and live long lives in places where food drifts by rarely. Their success depends on precise balance rather than mobility. From internal cooperation, attention shifts to environments where chemistry creates physical boundaries so extreme that even water itself forms barriers deadly to most animals. Brine pools form when extremely salty water collects in depressions on the seafloor. Because the brine is denser than surrounding seawater, it creates a distinct boundary that looks like a shimmering shoreline underwater. Crossing into a brine pool can be lethal for most animals. Oxygen is scarce and salinity is extreme, yet life still exists around the edges. Specialized microbes thrive on chemical gradients, and some animals patrol the margins to feed. Marine biologists study brine pools to understand how chemistry shapes habitat as clearly as physical terrain. These pools create invisible walls defined by density rather than stone. Brine pools demonstrate that the ocean contains environments within environments, layered and separate. From these chemical lakes, attention shifts to animals that survive cold, darkness, and time itself, building fragile structures that persist for centuries without sunlight. Below the photic zone, sunlight no longer provides energy, yet life continues. In these darker regions, food arrives from above as drifting particles, sinking remains, and occasional large falls like dead animals. This slow rain of material supports communities that live with patience rather than abundance. Many animals here move deliberately, conserving energy. Bodies are often soft, flexible, or elongated, suited to low-light conditions where speed is less useful than efficiency. Some species rely on chemical cues or vibration instead of vision, while others create light of their own to communicate or lure prey. The absence of sunlight also shapes time itself. Days blur together, seasons weaken, and life follows rhythms tied more to currents than to clocks. These dark zones remind marine biologists that sunlight is not the only driver of ecosystems. To understand how energy still enters these depths, attention turns to the smallest drifting organisms that quietly feed nearly everything else. Plankton are small organisms that drift with ocean currents, and despite their size, they support nearly all marine life. They include phytoplankton, which use sunlight to grow, and zooplankton, which feed on them and pass energy upward through the food web. Though often invisible individually, plankton blooms can stretch across vast regions. Their abundance rises and falls with light, nutrients, and water movement. When conditions are right, they multiply quickly, forming the first link between the sun and animals that swim, hunt, and migrate. Plankton are also travelers. Carried by currents, they connect distant ecosystems and transport energy across the ocean. Many animals begin life as planktonic larvae, drifting before settling into adult habitats. This drifting stage blends survival with chance, shaping population connections across entire basins. As plankton feed others, they also quietly influence the air above the sea, drawing attention next to their role in producing oxygen itself. A large portion of the oxygen in Earth's atmosphere is produced by marine phytoplankton. As they photosynthesize, these microscopic organisms release oxygen as a byproduct, quietly contributing to the air breathed by animals on land and sea alike. Unlike forests, plankton do not stay in one place. They bloom, fade, sink, and regrow, responding quickly to changes in light and nutrients. This constant turnover makes ocean oxygen production dynamic rather than fixed. Some regions pulse with productivity while others remain sparse. This process links the ocean to climate. When plankton grow, they absorb carbon dioxide, temporarily storing it in living tissue. Some of that carbon later sinks into deeper waters, removed from the atmosphere for long periods. Oxygen production and carbon storage are woven together in this exchange. Understanding this leads naturally to the movement of water itself, because currents decide where plankton thrive and where their influence travels next. Ocean currents move heat, nutrients, and life around the planet like slow, invisible rivers. Driven by wind, temperature, salinity, and Earth's rotation, these flows connect distant regions that may never touch by land. For marine organisms, currents can mean opportunity or risk. Plankton drift along them, larvae travel vast distances, and migrating animals follow predictable paths shaped by moving water. A change in current patterns can alter entire ecosystems, shifting where food appears and where animals gather. Currents also regulate climate by transporting warm water away from the tropics and cooler water back toward the equator. This redistribution moderates temperatures on land and influences weather patterns across continents. Beneath the surface, deeper currents move slowly but steadily, carrying cold, dense water across ocean basins over centuries. These layered flows create a constantly moving framework that life must adapt to. Within this motion, animals evolve strategies for travel, sensing, and timing, leading into the remarkable migrations that define many marine species. Deep-sea corals live far below sunlight in cold, dark waters. Unlike tropical corals, they do not rely on photosynthetic algae. Instead, they capture drifting food particles from passing currents. These corals grow very slowly, sometimes adding only tiny increments each year. Over centuries, they form branching structures that provide habitat for fish and invertebrates on otherwise bare sea floor. Marine biologists study deep-sea corals because they are both ancient and vulnerable. A single disturbance can destroy structures that took hundreds of years to build. Their skeletons also record chemical information about past ocean conditions. Though hidden, these corals shape deep ecosystems quietly. They are landmarks in darkness, offering shelter and stability. From long-lived corals, attention moves to animals that may live even longer, growing slowly in deep water and filtering the ocean itself over spans that exceed human history. Some deep-sea sponges may live for hundreds or even thousands of years. In cold, stable environments, their slow growth allows remarkable longevity. Layer by layer, they build bodies that persist through centuries of change above. Sponges filter vast amounts of seawater over time, removing particles and recycling nutrients. Their bodies provide shelter for smaller animals, creating microhabitats within their porous structure. Marine biologists study these sponges to understand resilience and deep-sea stability. Their growth rates offer clues about how slowly ecosystems can operate when conditions remain constant. Because they grow so slowly, damage can be permanent on human timescales. Disturbance removes centuries of accumulation instantly. From ancient living filters, focus shifts to the ocean floor itself, which records history in a different way, storing past climates and biological change in layers of sediment beneath the living sea. The ocean floor preserves history in layers of sediment that accumulate over time. Dust, volcanic ash, and microscopic shells drift down and settle, forming a slow record of changing oceans. Scientists extract sediment cores to read this history. Chemical signatures and fossil remains reveal past temperatures, ice coverage, productivity, and oxygen levels. Each layer marks a different chapter. Marine biologists and oceanographers use these records to understand how ecosystems responded to previous climate shifts. This long view helps distinguish natural variation from rapid modern change. The Seafloor Archive shows that oceans have reorganized many times, sometimes gradually, sometimes abruptly. Life adapts, migrates, or disappears depending on pace. From stored history, attention returns to living systems that actively shape climate today through daily biological processes that move carbon and energy between surface and depth continuously. The biological pump describes how ocean life moves carbon from the surface into deep waters. Phytoplankton absorb carbon dioxide as they grow. When they die or are eaten, waste and remains sink as marine snow. This sinking material transports carbon into deeper layers, where it can remain isolated from the atmosphere for hundreds or thousands of years. This process quietly stabilizes Earth's climate. Not all carbon reaches the bottom. Much is recycled along the way, feeding mid-water ecosystems. Still, the portion that sinks deep matters on a planetary scale. Marine biologists study the biological pump because it links tiny organisms to global climate regulation. It operates continuously, driven by countless small lives. Understanding this process reveals how the ocean acts as a buffer against rapid atmospheric change. From carbon storage, attention moves toward human influences that threaten to disrupt these long-running systems in ways biology alone cannot counter. Most animals move through life in one direction. They are born, they grow, they reproduce, and they die. A small number of jellyfish break this pattern in an unusual way. Under stress, they can reverse their development, returning from a mature stage back into a younger form. This process is not immortality. predators, disease, and injury still matter, yet it allows the animal to repeat parts of its life cycle when conditions become unfavorable. Cells reorganize, tissues change roles, and development begins again. Marine biologists study this ability because it challenges assumptions about aging. It shows that cellular identity can remain flexible under certain conditions. Understanding how cells remember earlier states may inform broader biology. In the ocean, where environments can change quickly, flexibility can be a powerful survival tool. From animals that rewind time, attention shifts to others that survive danger not by retreating or reversing, but by sacrificing part of themselves and rebuilding it slowly afterward. Sea cucumbers appear slow and defenseless, yet some species possess a dramatic survival strategy. When threatened, they can expel parts of their internal organs through the body wall, leaving predators distracted by the discarded tissue. This response is costly. Losing organs reduces feeding ability and requires significant energy to recover. Over time, however, the sea cucumber can regenerate what was lost, rebuilding complex tissues without disrupting overall body function. Marine biologists study this process to understand regeneration and immune response. The animal must heal, regrow, and resume normal activity without infection or structural failure. Beyond defense, sea cucumbers play an important ecological role. As they feed, they process seafloor sediments, recycling nutrients and influencing sediment chemistry. Their slow movement masks quiet influence. This combination of vulnerability and resilience highlights how survival does not always depend on speed or strength. From regeneration and sacrifice, attention turns to animals that rely instead on intelligence and exploration to navigate complex seafloor environments. An octopus explores the world in a way humans do not. Its arms are covered with sensors that detect chemicals, allowing it to taste as it touches. When an octopus reaches into a crevice, it is sampling the environment before anything reaches its mouth. This ability is especially useful in habitats full of hiding places. Crabs, snails, and other prey often wedge themselves into cracks. Vision alone is not enough. Chemical sensing guides decision-making arm by arm. Each arm can act semi-independently, yet remains coordinated by a central brain. Marine biologists study this distributed nervous system to understand alternative forms of intelligence. Octopus behavior reflects curiosity, problem-solving, and learning. They manipulate objects, remember solutions, and adapt to changing situations. In an environment where shells, rocks, and shadows dominate, touch and taste merge into one sense. From tactile intelligence, focus shifts to animals that communicate visually, turning their entire bodies into changing messages written in light and color. Squid communicate using rapid changes in color and pattern across their skin. Specialized cells expand and contract to produce flashes, ripples, and bands that move faster than many predators can track. These displays serve multiple purposes. Squid use them during courtship, rivalry, coordination, and threat response. A single animal can send different signals on different sides of its body at the same time. Marine biologists study these displays to decode meaning from motion. Patterns often appear alongside specific behaviors, suggesting intentional signaling rather than random change. Color communication is especially useful where sound may not travel cleanly or where silence is safer. Light and contrast become immediate and precise tools. This visual language also supports camouflage. The same system that broadcasts information can erase the body's outline in an instant. Squid balance expression and concealment continuously. From communication written on skin, attention moves to animals whose social systems depend less on color and more on dramatic biological shifts that stabilize group structure itself. In some marine species, reproduction is shaped by social structure rather than fixed roles. Group composition determines who breeds and when. This is especially common in reef fish living in stable hierarchies. Clownfish provide a clear example. A group contains a dominant female, a breeding male, and several smaller non-breeders. If the female dies, the largest male physically transitions into a female. Another fish then becomes the breeding male. This change involves hormonal shifts and reorganization of reproductive organs over time. Marine biologists study this process to understand how social cues trigger biological transformation. Such systems ensure reproductive continuity when mates are scarce. Instead of searching for new partners, the group reorganizes itself. This flexibility reveals how evolution favors stability over rigid design. From socially guided bodies, attention shifts to animals whose survival depends less on group structure and more on extreme endurance beneath the surface. Penguins are birds adapted for life beneath the waves. Their wings evolved into stiff flippers that provide powerful propulsion underwater. In water, penguins move with precision and speed that contrasts sharply with their waddling gait on land. Their feathers trap air, providing insulation in cold seas. That trapped air also affects buoyancy, helping penguins rise and sink efficiently while diving. Some species hunt at impressive depths for extended periods. Marine biologists study penguins to understand hunting in a three-dimensional environment. Fish move up, down, and sideways, requiring constant adjustment. Penguins respond with agility rather than pursuit speed alone. Different species specialize in different waters, from icy Antarctic seas to temperate coasts. Each design reflects local conditions. From underwater flight, attention turns to mammals that share the sea but manage air, rest, and danger in very different ways while remaining alert even during sleep. Some seals can sleep using only one half of their brain at a time, a process called unihemispheric slow-wave sleep. This allows the other hemisphere to remain alert, monitoring breathing, posture, and surroundings. This behavior is especially common in fur seals when they are at sea. One eye may remain open while the opposite brain hemisphere stays active. The sides alternate over time, allowing rest without full vulnerability. Marine biologists study this adaptation to understand how sleep can remain flexible. On land, seals may sleep more deeply, adjusting based on risk. Unihemispheric sleep supports survival in environments where drifting, drowning, or predation are constant concerns. It allows rest without surrendering awareness. From adapted sleep, attention shifts to animals that manage breath and oxygen over long periods, pushing mammalian physiology to its limits during deep and extended dives. Whales dive deep on a single breath, entering cold, dark water where pressure and oxygen scarcity challenge survival. Their bodies respond by slowing heart rate and redirecting blood flow toward vital organs. Oxygen is stored in blood and muscle rather than lungs alone. This reduces nitrogen absorption and supports long dives without breathing. Some species can remain submerged for over an hour. Marine biologists study these adaptations using tags that record depth, movement, and sound. Data reveal how whales balance energy, oxygen, and pressure precisely. These dives are not reckless. They are controlled descents shaped by evolution. Each movement conserves oxygen while enabling hunting in deep water. Understanding diving physiology explains why disturbance matters. Sudden stress can disrupt carefully managed systems. From breath and pressure, attention turns toward animals that influence ecosystems not through hunting alone, but by redistributing nutrients across vast areas of the ocean. Whales feed at depth and release waste near the surface, transporting nutrients upward in a process sometimes called the whale pump. These nutrients fertilize surface waters where plankton grow. Plankton blooms support food webs that feed fish, seabirds, and other marine life. This means whales contribute to productivity beyond their role as predators. Marine biologists study this effect to understand how large animals shape ecosystems chemically. Nutrient movement influences where life flourishes. This process links deep feeding grounds to sunlit waters, connecting layers of the ocean through behavior rather than currents alone. Waste becomes a resource rather than an endpoint. Whales also move nutrients horizontally as they migrate, redistributing productivity across regions. From nutrient movement by animals, attention turns to smaller organisms that shape chemistry even more quietly, operating at scales invisible to the eye but immense in total effect. Marine microbes include bacteria, archaea, and microscopic algae that drive ocean chemistry. Though individually tiny, together they regulate nutrient cycles, oxygen production, and carbon storage. Microbes break down organic material, recycle nutrients, and form the base of many food webs. Some fix nitrogen, making it available to other organisms. Others influence acidity and trace chemistry. Marine biologists study microbes using genetic tools because many cannot be grown in labs. Water samples reveal communities that change rapidly with conditions. Microbial activity responds quickly to temperature, nutrients, and circulation. Small shifts can ripple outward, affecting larger organisms and ecosystem balance. Though unseen, microbes operate continuously. They shape the ocean's chemistry minute by minute, supporting everything from plankton to whales. From invisible workers, attention naturally moves toward equally small particles created by humans, whose influence on marine systems is growing and persistent. Or when plastic breaks into tiny fragments, it begins to behave like food. These microplastics drift through the sunlit ocean where plankton feed. Because many plankton species consume particles based on size rather than composition, plastic can be taken in accidentally. Once ingested, microplastics occupy space without providing energy. For small organisms living on narrow energy margins, this can reduce growth and survival. Microplastics can also carry chemicals on their surfaces, transferring them into living tissues. Marine biologists study this process because plankton form the base of many food webs. What enters at this level can move upward into fish, seabirds, and marine mammals. The effects may accumulate gradually rather than causing immediate collapse. Tracking microplastics is difficult because they vary in shape, size, and chemistry. Still, their presence is widespread. From particles mistaken for food, attention moves upward to animals that mistake larger floating plastics for prey, with consequences that unfold far above the water's surface. Many seabirds feed by scanning the ocean surface for floating prey. This strategy makes them vulnerable to plastic debris that resembles fish eggs, squid, or small fish. Some plastics also acquire the scent of ocean life, making them even more deceptive. When seabirds swallow plastic, the danger is not only choking. Plastic can fill the stomach, reducing hunger and limiting intake of real food. Adults may survive longer, but chicks fed plastic by mistake can suffer severe growth failure. Marine biologists study seabird ingestion to map debris hotspots and understand feeding behavior. Patterns often reveal links between plastic concentration and ocean circulation. This problem highlights how human materials intersect with evolved instincts. A bird adapted for precision can be undone by false signals drifting at the surface. From mistaken meals above the waves, attention shifts back below, to fishing tools lost at sea that continue catching animals without intention or restraint. Fishing nets and lines are designed to catch animals. When they are lost or abandoned at sea, they can continue trapping life indefinitely. This phenomenon is known as ghost fishing. Lost gear can drift, snag on reefs, or settle on the seafloor. Fish, crabs, and even large animals may become entangled. As trapped animals die, they attract scavengers, which can also be caught, creating a cycle that repeats. Marine biologists and divers document ghost gear to assess damage and guide removal efforts. Recovering nets is dangerous but can immediately reduce harm to a habitat. Ghost fishing is not simply litter. It is an active threat that keeps functioning long after it leaves human hands. From persistent traps, attention turns to another invisible pressure humans add to the sea, one that spreads through water as vibration rather than material. Sound travels efficiently through seawater, and many marine animals rely on it to navigate, find mates, and stay connected. Whales, dolphins, and some fish depend on specific frequency ranges for communication. Human activities add noise to these same ranges. Shipping, construction, and some sonar sources increase background sound levels. This can reduce the distance over which calls are heard, similar to trying to speak in a crowded room. Marine biologists study noise by combining acoustic recordings with animal tracking. Some species respond by calling louder or changing timing, which requires extra energy. Others avoid noisy areas entirely. Noise does not injure tissues directly, yet it alters behavior and movement patterns. Over time, this can reshape habitat use. From masked voices, attention moves to another danger near the surface, where large vessels and large animals sometimes intersect with fatal results. Whales must surface to breathe, often in areas close to shore where shipping traffic is dense. Large vessels move quickly and can be difficult to maneuver, especially in poor visibility. A collision between a ship and a whale can cause severe injury or death. Some species are especially vulnerable because they feed or rest near the surface for extended periods. Marine biologists assess risk by mapping whale distribution alongside shipping routes. These overlap zones reveal where danger is highest. Solutions can be practical. Reducing ship speed lowers collision risk and impact severity. Adjusting routes during sensitive seasons can also help. Ship strikes show how modern infrastructure intersects with ancient migration paths. Avoiding harm often depends on timing rather than exclusion. From collision risk, attention turns toward fishing practices, where small changes in design can significantly reduce unintended harm to marine life. Bycatch refers to animals caught unintentionally during fishing. Sea turtles, seabirds, and non-target fish species are often affected. Reducing bycatch is a major focus of modern fisheries science. Marine biologists work with fishers to test gear modifications. Certain hook shapes make it harder for turtles to swallow bait. Weighted lines help hooks sink faster, reducing bird interactions near the surface. In some fisheries, lights on nets increase visibility, allowing animals to avoid entanglement. These changes are designed to preserve catch while minimizing harm. Effective solutions must work in real conditions. A design that slows fishing too much will not be adopted. Success depends on cooperation and evidence. From smarter tools, attention shifts to broader protections that limit extraction entirely, giving ecosystems space to rebuild through time and reduced pressure. Marine protected areas restrict or prohibit certain activities within defined boundaries. When well designed and enforced, they can allow ecosystems to recover from overuse. Within protected zones, animals often grow larger and more abundant. Predators return, reshaping prey behavior and restoring balance. These changes can stabilize habitats over time. Marine biologists monitor protected areas using surveys, tagging, and underwater cameras. Recovery is measured across years, not weeks. Benefits can extend beyond boundaries. Fish and larvae may move outward, supporting nearby fisheries. Protection becomes a source rather than a barrier. Protected areas are not static. They require enforcement and adaptation as conditions change. From protection at scale, attention turns to damaged reefs specifically and the conditions under which they can slowly begin to rebuild their living structures again. A coral reef can recover if key conditions return. Clear water, stable temperature, and healthy populations of herbivores are especially important. Herbivorous fish and urchins keep fast-growing algae from covering open surfaces. Without them, algae can block light and space, preventing young corals from settling. Marine biologists look for early recovery signs such as coral larvae attaching to bare rock and increased diversity of reef fish. These small changes signal potential long-term improvement. Recovery is not guaranteed. Repeated stress can overwhelm resilience. Yet when pressures ease, reefs can surprise researchers with persistence. This process highlights how ecosystems depend on interactions rather than single species. From natural recovery, attention shifts to active restoration efforts where humans assist rebuilding by working with coral growth rather than against its pace. Some coral restoration projects begin with small fragments grown in nurseries. These nurseries may be underwater or in controlled tanks on land. Fragments are carefully tended and multiplied. Once grown, corals are transplanted onto damaged reefs and secured. Marine biologists track survival, growth, and resistance to stress. Placement and water quality strongly influence success. Restoration is not a shortcut. Corals grow slowly and restored reefs remain vulnerable if surrounding conditions remain poor. Success depends on reducing ongoing stressors. Still, nurseries offer hope. They work with coral biology rather than forcing rapid change. Each fragment represents patience. From rebuilding reefs piece by piece, attention turns to coastal habitats facing a different challenge, where rising water leaves little room for ecosystems to retreat. As sea level rises, coastal habitats such as marshes and mangroves attempt to move inland. In many places, roads, walls, and buildings block this movement. The result is coastal squeeze. Water advances from one side while hard infrastructure resists from the other. Habitats become narrower each year, even if conditions remain otherwise suitable. Marine biologists map this process by comparing historical and modern shoreline data. Loss of space reduces nursery habitat for fish and weakens natural storm protection. Coastal squeeze is a problem of geometry as much as climate. Without room to shift, ecosystems can disappear rather than migrate. From shrinking edges, attention moves toward broader climate-driven changes that rearrange marine life itself, shifting species ranges, timing, and interactions across entire ocean basins. Many marine species live within narrow temperature ranges. As ocean waters warm, those comfort zones move, often towards the poles or into deeper water. Fish may appear in regions where they were once rare, while others retreat from familiar coasts. Marine biologists track these shifts using surveys, catch records, and tagging data. Changes can happen gradually or arrive suddenly during marine heatwaves that open temporary corridors for movement. These shifts affect more than location. Timing can change as well. Spawning seasons may move earlier, migrations can lengthen, and predator-prey relationships can be rearranged. A new arrival may compete with resident species or become prey for predators unaccustomed to it. Range shifts show how quickly ecosystems can reorganize when temperature changes. They are not simple migrations from one place to another. They are rewiring events that alter food webs and energy flow. From warming-driven movement, attention turns to another subtle chemical change unfolding alongside temperature rise, one that quietly alters how shells and skeletons are built. As the ocean absorbs carbon dioxide from the atmosphere, seawater chemistry changes. This process is known as ocean acidification. It reduces the availability of carbonate ions needed by many organisms to build shells and skeletons. Shell builders such as oysters, mussels, some plankton, and corals can be especially vulnerable. Early life stages are often most affected as tiny larvae struggle to form their first protective structures. Marine biologists study acidification by measuring water chemistry and observing growth and survival under different conditions. In some regions, natural upwelling brings deeper, more acidic water to the surface, intensifying effects seasonally. Acidification does not dissolve shells overnight. It acts more like resistance against construction, making growth slower and more energetically costly. From chemical headwinds against growth, attention shifts to biological responses that can unfold quickly, sometimes explosively, when conditions briefly favor certain microscopic organisms. Most algae are normal and beneficial parts of ocean ecosystems. However, certain species can bloom in ways that harm wildlife and people. These harmful algal blooms occur when conditions align, such as warm water, abundant nutrients, and calm seas. Some blooms produce toxins that accumulate in shellfish and move up food chains. Others create dense mats that block light and reduce oxygen when they decay. Fish kills and shellfish closures can follow. Marine biologists monitor blooms using water samples, satellites, and coastal surveys. Blooms can shift location with currents, appearing far from where they began. The unsettling aspect is speed. A coastline can seem healthy one week and face closures the next. Blooms reflect how quickly biology responds to opportunity. From blooms that deplete oxygen locally, attention moves toward broader changes in oxygen distribution that reshape where marine animals can survive at all. Oxygen is unevenly distributed in the ocean. In some regions, mid-water layers contain very little oxygen, forming zones where many animals struggle to survive. These areas can expand as ocean temperatures rise. Warmer water holds less dissolved oxygen. Changes in circulation and productivity can also reduce replenishment. As low oxygen zones grow, animals may be forced into thinner surface layers where oxygen remains available. Marine biologists study oxygen levels using sensors on ships, floats, and autonomous vehicles. They link these measurements to animal movements and fisheries data. Crowding near the surface can increase competition and predation. Less mobile organisms may suffer most because they cannot easily escape. From oxygen limits on life, attention shifts to large climate patterns that rearrange currents and nutrients across entire ocean basins for months at a time. El Niño events occur when surface waters in the Pacific Ocean warm and wind patterns shift. These changes alter nutrient upwelling, reducing productivity in some regions while increasing it elsewhere. When the base of the food web changes, everything above it feels the effect. Fish may move, seabirds may struggle to find food, and breeding success can rise or fall dramatically. Marine biologists study El Niño as a natural experiment. It reveals how sensitive ecosystems are to temperature and circulation changes and how quickly they respond. El Niño does not affect all regions equally. Some coasts experience warmer, clearer water, while others face increased rainfall and runoff that influence coastal habitats. From seasonal reshuffling, attention turns to sudden disturbances that strike locally, reshaping reefs and coastlines in single dramatic events. Powerful storms can break corals, topple structures, and stir sediments that cloud the water. In a single night, years of growth can be damaged or lost. Yet storms can also deliver nutrients by mixing deeper water upward and washing material from land into coastal seas. Afterward, productivity may temporarily increase. Marine biologists study storms as disturbances with two faces. They measure immediate damage, then track recovery over months and years. Outcomes depend on timing and existing conditions. A healthy reef may rebound. A stressed one may shift toward algae dominance instead. Storms highlight how change in the ocean is not always gradual. Sometimes it arrives suddenly, setting the stage for new ecological trajectories. From episodic disturbance, attention turns to large animals that influence ecosystems even without storms, redistributing nutrients simply by feeding and surfacing. When whales feed at depth and surface to breathe, they release waste near the surface. This waste contains nutrients that fertilize phytoplankton, supporting primary production. This process, sometimes called the whale pump, links deep feeding zones with sunlit waters where photosynthesis occurs. Nutrients that might remain deep are returned upward. Marine biologists study this effect to understand how large animals contribute to ecosystem productivity. Whales are not just consumers. They are movers of materials. Migrating whales also transport nutrients horizontally across regions, influencing productivity far from feeding grounds. From nutrient movement by animals, attention shifts to predators whose influence is less about chemistry and more about shaping behavior through presence and risk. Top predators influence ecosystems not only by consuming prey but by changing how prey behave. The presence of a predator can alter where and when prey feed. Fish may avoid certain areas or spread grazing pressure more evenly. These behavioral changes can protect habitats such as seagrass beds or reefs from overuse. Marine biologists study these effects by tracking movement and observing habitat use. The results show that fear can shape ecosystems as powerfully as hunger. When predators decline, prey may become bolder. Overgrazing or overpredation can follow, leading to habitat loss and instability. From invisible choreography of risk, attention turns to a well-known example where one predator's return reshaped an entire underwater forest. In some coastal regions, sea urchins graze intensely on kelp, stripping reefs down to bare rock. These areas are known as urchin barrens. Sea otters hunt urchins, reducing their numbers and changing grazing behavior. When otters are present, kelp forests can recover and grow dense. Marine biologists describe sea otters as a keystone species because their impact far exceeds their numbers. By eating one prey, they shape an entire habitat. Kelp forests provide shelter, food, and nursery space for many species. They also slow waves and influence coastal chemistry. From predator-driven forests, attention shifts to new tools that allow scientists to observe marine life across vast scales previously impossible to monitor. Advances in satellite imaging allow researchers to detect whales near the ocean surface from orbit. Under calm conditions, whale-sized shapes can be identified in high-resolution images. Marine biologists use repeated satellite passes to estimate abundance and seasonal patterns, especially in remote regions difficult to survey by ship. This method does not replace fieldwork. Satellites cannot easily determine species, age, or health. Still, they expand the scale of observation dramatically. From orbit, the ocean becomes a map of movement and presence rather than an empty expanse. Patterns emerge that guide focused research on the water. From eyes in space, attention turns toward techniques that detect life without sight at all, using traces left behind in the water itself. Every organism in the ocean leaves traces behind. Scales, mucus, waste, and tiny fragments of tissue carry genetic material into the water. Environmental DNA, often called eDNA, allows scientists to detect species by sampling seawater alone. Marine biologists collect water, filter it, and analyze genetic sequences to identify which species have recently been present. This method can reveal elusive animals that hide, travel at night, or live in difficult environments. eDNA does not show exactly how many individuals are present, and currents can move genetic traces. Signals also fade over time. Still, the technique is powerful for early detection of rare or invasive species. By listening for biological signatures rather than sightings, scientists expand their reach. Entire communities can be inferred without nets or cameras. From invisible genetic traces, attention moves to tools that follow individual animals directly, recording their journeys through depth, temperature, and time in remarkable detail. Modern tracking tags have become small enough to follow animals once impossible to study. These devices can record depth, temperature, movement, and sometimes light levels that help estimate location. When a tagged animal is recaptured or the tag detaches and floats to the surface, the stored data reveal hidden routines. A fish may dive deeper during the day and rise at night, following temperature layers rather than landmarks. Marine biologists use these records to understand habitat use and migration paths. This information guides conservation by revealing where animals actually spend time. Tag data often show complex behavior rather than straight-line travel. Movements respond to currents, comfort zones, and opportunity. From individual journeys mapped by sensors, attention turns to a broader sensory world where sound reveals presence even when animals remain unseen. Underwater microphones called hydrophones allow scientists to hear the ocean. Reefs crackle with snapping shrimp. Fish produce hums and knocks. Whales send calls that travel vast distances. Marine biologists deploy hydrophones for weeks or months, building sound records that reveal daily and seasonal patterns. A reef may sound different at dusk than at midday. Migration can announce itself acoustically before animals appear. Soundscapes act as fingerprints for habitats. Different communities create different acoustic signatures. Changes in sound can indicate shifts in biodiversity or rising human noise. Listening complements visual surveys, especially in murky or deep water. Sound travels where light fails. From acoustic worlds, attention moves toward machines that explore places humans cannot, mapping terrain and life in darkness and pressure. Autonomous underwater vehicles, or AUVs, can map the seafloor in detail. They travel close to the bottom, using sonar and cameras to scan landscapes hidden from surface ships. These robots navigate ridges, valleys, and steep slopes, following programmed paths without direct control. Some operate for long periods before returning with data. Marine biologists use AUV maps to locate habitats such as sponge gardens, coral mounds, and seamount communities. Texture and shape often predict where life gathers. A smooth plain may hide burrows. A rocky rise may host unexpected diversity. Mapping is the first step toward understanding distribution. From robotic scouts, attention shifts to discoveries made when scientists look even closer, finding life not just on rocks, but within them. The seafloor appears solid from a distance, but up close it is full of cracks, pores, and fissures. These tiny spaces create microhabitats where water flows and food particles collect. Marine biologists using submersibles have found animals living inside these cracks. Small filter feeders benefit from steady currents. Grazers scrape microbial films from stone surfaces. Living inside rock offers protection from predators and strong currents. It also provides stable conditions in otherwise harsh environments. This discovery expands the definition of habitat. It is not only reefs and mud, but architecture hidden within stone itself. From life in cracks, attention moves to organisms that build delicate structures in cold darkness, shaping deep ecosystems over extraordinary spans of time. Some deep-sea corals grow for centuries in cold, dark water. As they build skeletons slowly, they incorporate chemical information from surrounding seawater. Marine biologists study coral skeletons to reconstruct past ocean conditions. Isotopes and trace elements reveal changes in temperature, circulation, and chemistry over time. These corals provide habitat for fish and invertebrates, forming fragile frameworks on the seafloor. Their slow growth makes them vulnerable to disturbance. A single impact can erase centuries of structure. Recovery, if possible, takes far longer than a human lifetime. From coral historians, attention turns to even older organisms that persist quietly, filtering water and witnessing changes across millennia. Some sponges in deep, cold environments may live for thousands of years. Their slow growth allows extreme longevity under stable conditions. Sponges filter enormous volumes of water, removing particles and recycling nutrients. Over centuries, this filtration influences local water chemistry and clarity. Their bodies provide shelter for small animals that live within channels and folds. A sponge becomes a structure as much as an organism. Marine biologists study long-lived sponges to understand resilience and growth limits. They also use growth layers to infer past environmental change. Because sponges grow slowly, damage is effectively permanent on human timescales. Disturbance removes accumulated history instantly. From ancient filters, attention moves to another hidden seafloor feature that stores energy, risk, and unique ecosystems all at once. Methane hydrates form when methane molecules become trapped inside cages of frozen water under high pressure and low temperature. They resemble ice but burn when warmed. These deposits exist in some seafloor sediments and represent vast stores of methane. Marine scientists study them for their role in climate and seafloor stability. If hydrates destabilize, methane can escape, influencing greenhouse gas levels. Sediments may weaken, increasing landslide risk. Hydrates are also linked to seep ecosystems. Methane escaping slowly can support microbes and animals that use chemical energy. From chemical ice, attention shifts to extreme environments where dense water forms deadly boundaries and life gathers at the edge. Brine pools are underwater lakes of extremely salty water that settle into seafloor depressions. Because the brine is denser than surrounding seawater, it forms a sharp boundary. Crossing this boundary can be fatal for many animals due to low oxygen and extreme salinity. Yet life thrives nearby. Microbes feed on chemical gradients at the edges. Marine biologists study brine pools as examples of chemistry shaping habitat. The boundary acts like an invisible wall. Some animals patrol the margins, feeding where conditions exclude competitors. Life clusters where chemistry allows opportunity. From chemical boundaries, attention turns to organisms that tolerate extreme heat, redefining where life can exist at all. Hydrothermal vents release superheated fluids into icy deep water, creating steep temperature gradients. Some animals live astonishingly close to these heat sources. Vent worms, clams, and microbes rely on chemosynthesis rather than sunlight. Proteins and enzymes are adapted to function under extreme conditions. Marine biologists study these systems to understand the limits of life. Vents show that heat and chemistry can support ecosystems without solar energy. Vent fields appear and disappear over time. Species must disperse and colonize new sites, making survival dependent on timing and chance. From heat-fueled life, attention naturally turns toward the deepest regions of the ocean, where pressure becomes the defining force shaping all remaining inhabitants. At the deepest parts of the ocean, pressure becomes the dominant force shaping life. In trenches such as the Mariana Trench, pressure exceeds 1,000 times that at the surface. Most surface animals would be crushed instantly, yet specialized organisms persist. These animals are not held together by pressure. Instead, their bodies are composed largely of water and fluid-filled tissues, allowing internal pressure to match the surrounding environment. They lack gas-filled spaces that would collapse under force. Marine biologists study trench life using landers and submersibles built to withstand immense pressure. The animals discovered are often small, slow-moving, and adapted to extreme scarcity. Food arrives only as drifting material from far above. Energy is conserved carefully. Metabolism runs slowly. Patience becomes survival. Trench ecosystems remind scientists that biology does not resist physics. It works within it. From survival under pressure, attention shifts to body designs shaped not by strength, but by softness and buoyancy in the deep sea. Many marine animals begin life as larvae that drift freely in the water. During this stage, they may look nothing like their adult forms. existing instead as tiny, fragile travelers carried by currents. Larval drift spreads species across wide areas, connecting reefs, coasts, and open ocean habitats. It also introduces uncertainty. A larva must survive predators, find food, and eventually encounter the right conditions to settle and transform. Many never do, making survival partly a numbers game. This drifting phase allows populations to recover after disturbance. If one area is damaged, larvae from elsewhere can recolonize it. At the same time, it means local conditions far away can influence nearby ecosystems. Marine biologists study larval movement to understand resilience, connectivity, and long-term stability. From drifting larvae, life grows into complex forms that must sense, hunt, and avoid danger. To do so in a vast, moving environment, many marine animals rely on senses that differ from those we use most on land. In water, vision is often limited by distance, darkness, or suspended particles. As a result, many marine animals rely on other senses to understand their surroundings. Sound, vibration, smell, and electrical signals often matter more than sight. Fish use sensory lines along their bodies to detect pressure changes caused by movement nearby. Sharks and rays can sense weak electrical fields produced by muscle activity in other animals. Whales and dolphins rely heavily on sound, which travels efficiently through seawater and carries information across great distances. These senses create a world rich with signals humans rarely notice. A small shift in current, a distant call, or a faint vibration can guide behavior. Survival depends on reading this invisible information accurately. As sound becomes especially important, communication takes on new forms. From low whale calls to high-pitched clicks, the ocean becomes an acoustic landscape. That sound-based world leads directly to some of the most complex communication systems found among marine mammals next. Whales live in an ocean where sound can travel hundreds of miles under the right conditions. Low-frequency calls move efficiently through deep water, bending with temperature layers and reaching far beyond the horizon. These sounds serve many purposes. Whales use them to maintain contact, coordinate movement, and navigate through vast spaces. Some species produce patterned songs that can change slowly over time, spreading between groups like cultural traditions. Because light fades quickly underwater, sound becomes the primary way whales experience their world. It helps them locate food, avoid obstacles, and stay connected across distance. Marine biologists study whale acoustics using underwater microphones, building long-term records of movement and behavior. This sound-based life is sensitive to change. Noise from ships and industry can interfere with communication, shrinking the acoustic space whales depend on. Understanding this makes sound not just a curiosity, but a vital part of marine ecology, guiding attention next to animals that communicate in more personal, individualized ways. Dolphins live in dynamic social groups where individuals separate and reunite often. To stay connected, many dolphins use signature whistles, distinctive vocal patterns that function like individual identifiers. Each dolphin develops its own whistle early in life. Others can recognize and even imitate that sound to address the individual directly. This allows dolphins to maintain social bonds in an environment where visibility changes constantly. Signature whistles do not form a language in the human sense, but they reflect advanced social awareness. Dolphins remember individuals, track relationships, and coordinate behavior using sound. Marine biologists study these whistles by recording underwater calls and matching them to observed individuals. Because sound travels so well in water, a dolphin does not need to be nearby to be known. A familiar whistle can carry presence across distance. This reliance on sound also appears in fish communities where hearing and vibration shape interactions in quieter but equally important ways. Many deep-sea animals appear fragile and gelatinous, yet their softness is an advantage. In deep water, pressure is immense but evenly distributed. Water supports these bodies, eliminating the need for rigid skeletons. These animals are mostly composed of fluids that do not compress. Internal and external pressure remain equal, preventing collapse. Without gas-filled organs, there is nothing to implode. Marine biologists study these forms to understand alternative solutions to structure. Soft tissues allow expansion when feeding and require little energy to maintain shape. Movement can be slow and efficient. Gelatinous animals often drift rather than swim, conserving energy in a food-poor environment. Their transparency can also reduce visibility to predators. Softness here is not weakness, it is alignment with surroundings. From bodies shaped by pressure, attention moves towards sensory adaptations that allow animals to perceive a world without light, landmarks, or sound cues familiar to humans. In deep water, vision often becomes limited or useless. Many animals rely instead on touch, vibration, and chemical sensing to understand their surroundings. Sensory systems shift to match the environment. Some fish detect minute pressure changes through specialized sensory lines along their bodies. Others sense electrical signals generated by living tissue nearby. These cues reveal movement, proximity, and prey without sight. Marine biologists study these senses to understand how animals navigate darkness. Sensory input becomes slower but more precise. Reaction replaces anticipation. In this environment, information arrives quietly. A faint vibration can signal danger. A chemical trace can guide a predator. The deep ocean is not empty of information. It is rich in signals humans do not naturally perceive. Understanding these sensory worlds expands how scientists define awareness and interaction. From perception without sight, attention moves to communication systems that use sound, vibration, and timing to connect animals across vast distances underwater. Sound travels farther and faster in water than in air. Many marine animals depend on it as their primary sense for communication and navigation. Whales, dolphins, and fish produce calls that can travel miles. Low-frequency sounds can bend through layers of temperature and depth, reaching far beyond visual range. This allows animals to remain connected across open ocean spaces. Marine biologists record these sounds using hydrophones, revealing patterns linked to migration, feeding, and reproduction. Some calls repeat over years, changing slowly like traditions. Sound also shapes habitat use. Animals may avoid noisy regions or adjust timing to communicate when conditions are quieter. The ocean is filled with overlapping soundscapes. Some are natural, others introduced. Communication depends on clarity and timing. From sound as connection, attention turns to collective behavior, where individual actions scale into coordinated movement across thousands of bodies acting as one. Schools of fish move as if guided by a single mind, yet no leader directs them. Coordination emerges from simple rules followed by each individual. Stay close to neighbors, match direction, avoid collisions. When thousands follow these rules at once, the group flows, compresses, and turns in unison. Predators struggle to isolate a single target within shifting patterns. Marine biologists study schooling to understand how collective behavior arises. Models show how information spreads faster through a group than any single animal could react. Schools also function as sensors. A sudden turn by a few can ripple outward, warning the entire group of danger. This behavior shows how cooperation can arise without planning. Complexity grows from simplicity. From group movement, attention shifts to long-term patterns where life history unfolds slowly, measured not in seconds or days, but in centuries and geological time. Some marine organisms grow so slowly that their bodies record time itself. Deep-sea corals, sponges, and sediment-dwelling creatures accumulate layers over decades or centuries. Marine biologists read these layers like calendars. Chemical signatures reveal changes in temperature, productivity, and circulation. These organisms become living archives. Their longevity depends on stable conditions. Slow growth trades speed for endurance. Disturbance removes history instantly. These timekeepers connect present oceans to past states, revealing how systems responded to earlier change. They offer context for modern shifts. Life here is not rushed. It persists quietly, recording while existing. From organisms that store memory in structure, attention turns to the ocean as a whole, a system still largely unexplored, where discovery remains ongoing rather than complete. Despite centuries of exploration, much of the ocean remains unseen. Depth, darkness, distance, and pressure limit observation. Many species are known only from single specimens or fleeting glimpses. Every new tool tends to reveal more complexity rather than closure. Cameras capture unexpected behavior. Water samples reveal unseen communities. Maps uncover habitats previously hidden. Marine biologists understand the ocean not as a solved puzzle, but as a living system full of unanswered questions. Discovery often arrives quietly, without spectacle. The unknown is not empty space. It is crowded with interactions waiting to be understood. Curiosity continues to be rewarded. From mystery itself, the journey naturally slows. The ocean continues moving whether observed or not. Knowledge accumulates gradually, like sediment settling through deep water. And with that steady rhythm, the facts begin to rest. As the ocean continues its quiet work beneath the surface, you can let these thoughts drift and settle. The tides will keep moving, the currents will keep turning, and life will carry on in darkness and light alike. There is nothing more to hold now. Sleep gently.